Okay, so in our last class, we discussed about determination of structure of peptide or protein. So the what is called hydrolysis method only give the amount and number of amino acid present in that. So whereas the Sanger's method, so it will give you the what is the amino acid present in the N-terminal end, and the Headman method, it will give you the information about the sequence of amino acid present in a uh, uh, present in a peptide or protein. So these three methods are give you the information about the amino acids. That's more particularly Hedman method will give you the sequence of amino acid in which the peptide or protein is formed. And in terminal end, it is possible to find out by using a Sanger's reagent. Okay, that's called dinitrochlorobenzene is the Sanger's reagent. So for his invention, and one more thing, Sanger himself, he uh, prepared a compound called insulin, and he is the first person who is reported a peptide is having 18 amino acid unit. Let's give an entry. So for his work, Nobel Prize Committee given a Nobel Prize to Sanger for his contribution towards the peptide chemistry, more particularly the synthesis of insulin. So here, now we know that suppose a peptide or protein given. So if we are doing a hydrolysis, so we will come to know that amount of amino acid and the kind of amino acids are involved in a peptide or protein. Suppose that is subjected to a what is Sanger's reagent, we will come to know what is the N-terminal end of the amino acid. Suppose that is subjected to Hedman's reagent, we will come to know that sequence of amino acid involved in the peptide or protein. So this is about uh, determination of structure of peptide or protein using Sanger's method and Edmonds method and hydrolysis method. So with this, I will, so let's give an entry. So with this, I will go to the next concept. It's called cleavage of peptide bond. So you know that peptide is made up of amino acid. Amino acid contains amino group as well as carboxylic end. So amino group means acidic, sensitive to acidic. Carboxylic group means sensitive to basics. So that is the reason. Uh, let's give an entry. Right. So that is the reason how we can cleave the peptide bond. Suppose a peptide bond, it's here of two kinds. One is a chemical method, another one is enzymatic method. So in enzymatic method, I will explain about enzymatic method at the end of this class. And first I will take up the chemical method of cleavage of peptide bond. So under that, the first method is acidic hydrolysis. So let's give an entry. Right. Okay. So cleavage of peptide bond. These are two kind. One is a chemical method. Another one is enzymatic method. So since we are organic chemistry or chemistry point of view, we should discuss more about chemical methods. And enzymatic method also we will discuss, but not much more detail. So that is the reason I will discuss what is maybe at the end of this class. So first I will take up the chemical method. Under that chemical method, I will take up acidic hydrolysis. That means a peptide or protein, when it is subjected to hydrolysis, so more particularly 6 normal hydrochloric acid. I think whatever the laboratory, okay, laboratory means more particularly the origin, not in the table reagent. Table reagent will have a 2 normal one. So that means a little higher concentration to the table reagent 6 normal one. So say I think you know that 1 minute. Let's give an entry. Right. Okay. 6 normal. Okay. The commercially available in the laboratory, commercially available hydrochloric acid is around 11 to 12 normal, 12 normal. So, but that we have to reduce it to 6 to 12, 6 normal hydrochloric acid. Maybe by so what will happen? The peptide bond. So peptide bond present in a polypeptide or a peptide or a protein cleaves and leaving behind amino acid units. So after looking into the reaction, we will come to know that what and all the amino acids are present in that by doing here, as I mentioned that amino acid contain amino group as well as acidic carboxylic group. So amino group is sensitive to acidic, carboxylic acid is group is sensitive to basic. So that is, and also you know that acidic amino acid, basic amino acid, neutral amino acid. So, but when it is treated with acid, 6 normal HCl is an acid. So, in this, the drawback of this method is tryptophan. Okay, so tryptophan, cysteine. Cysteine is the one what is called as a sulfur-containing amino acid. Serine is the hydroxy-containing amino acid. Threonine is also a hydroxy-containing amino acid. Okay, these compounds, okay, tryptophan is a indole-containing amino acid. So all slowly destroyed. So slowly these compounds are slowly destroyed. And guanine and aspartic undergoes deamination, okay, giving rise to glutamate and aspartic acid. So glutamic acid undergoes intramolecular dehydration to give pyrrolidine 5-carboxylic acids. So these are the drawback of this acidic hydrolysis. So what is the advantage then? In spite of these difficulties, acidic hydrolysis is ordinarily the method of choice. So because amino acids are not racemized. So that means in this method, amino acids are not racemized. And one more thing, in this method, a tryptophan, cysteine, serine, threonine are slowly destroyed, and glutamine and aspartic acid are converted into glutamate and aspartate, and glutamic acid undergoes rearrangement, giving rise to dehydration, giving rise to pyrrolidine 5-carboxylic acid. So this is about acidic method. So let's go to the basic method. So let's see what are the advantages, what are the disadvantages of the basic hydrolysis method of cleavage of peptide. So here we are talking only about the cleavage of peptide. Right. So, alkaline hydrolysis. The complete hydrolysis of protein may also be carried out by heating them with 2 normal to 6 normal sodium hydroxide at 110 at 100 degrees centigrade for about 4 to 8 hours. So the drawback of this method is racemization of the amino acid occurs. So I think you know that only the L amino acids are physiologically active. So D1 is not physiologically active. Suppose if you are taking sodium hydroxide and cleaving the peptide means during this process a racemization. Suppose L is converting into a D means then its physiological activity is destroyed. So that means this is not the better method for cleavage of peptide. So then cysteine racemization is one problem. Another one is cysteine. Cysteine means sulfur containing amino acid. Serine means a CH2OH, a alcohol containing amino acid. Threonine also alcohol containing amino acid. Arginine is a heterocyclic containing amino acid. So these are destroyed when a peptide is cleaved by using alkaline hydrolysis. So then what is the advantage about this method? So in spite of these limitations, alkaline hydrolysis is useful for the analysis of tryptophan. So that means in this process, alkaline hydrolysis, tryptophan is stable. So we can identify the tryptophan amino acid presence by doing alkaline hydrolysis of a peptide or a protein. So because it's not destroyed during this process. Then one more thing. So as I mentioned earlier, amino acids are sensitive to acidic as well as the basic. So and one more thing, accurate determination of amino acid composition of a peptide or protein is great importance. So as I mentioned that L amino acid is active, physiologically active one. So if there is a difference in stereochemistry, L instead of L, D is there means then it won't be a physiologically active. So because of that, accurate determination of amino acid composition, and protein, okay, amino acid composition of a protein or a peptide is a great importance in their characterization and for understanding their structure. Such analysis involves two stages. So one is hydrolysis of a peptide or a protein, and second is separation of the hydrolyzed product. So in this connection, cleavage of peptide by using acidic hydrolysis, it will have certain disadvantages. By using alkaline hydrolysis, it will have certain disadvantages. That is the reason. So a special method of cleavage of peptides are involved, and here one chemical called what is called as hypobromide. Okay, hypobromide is used. So this hypobromide, what it will do? Hypobromide cleave. Okay, hypobromide cleave the C-terminal peptide. So you know that C-terminal end and N-terminal end. So normally any peptide when you are writing a peptide on a board or a paper, left hand side there will be a free amino group that is called N-terminal end. Right hand side there will be a free carboxylic group that is called C-terminal end. So what I am saying here, hypobromide means HOBr. So this HOBr is cleaves a peptide. Okay, here I am taking a one peptide, more particularly a peptide having this here. I am taking this is the 24th amino acid residue. This will be the 25th amino acid residue. This will be the 26th amino acid residue. It's a peptide I'm taking here. So this peptide at the 25th amino acid, the residue is tyrosine, isn't it? So what it means? So it means that hypobromide, it cleaves the peptide bond at the tyrosine and adjacent C-terminal end of the peptide. So that means it will cleave here. So this so that means it hypobromide cleaves the what you call specifically cleaves the C-terminal peptide. Specifically cleaves the C-terminal peptide bond. So between the tyrosine, this is the tyrosine, and adjacent amino acid, resulting in two fragments. I will show you. This is one fragment. This is another fragment. So it doesn't affect any other thing. It selectively cleaves the tyrosine and the adjacent amino acid of a peptide or a protein. So that is the speciality about hypobromide. Hypobromide means HO Br. So in this HO minus and Br plus. So what it will do? So for this explanation, I took a peptide is having a tyrosine at the 25th amino acid residue. So when this is treated with HOBr, so what will happen? There is a bromination. So where it will bromination? So this carbon is there, no? This carbon gets brominated. So then there should be a removal of proton from here. One proton from here, another proton from here. So 2 molecule of HOBr. So giving rise to 2 molecule of water is going out. There will be a bromine here, there will be a bromine here. So in the first step, there is a formation of dibromo compound. So it's a electrophilic substitution reaction. So this ortho position of hydroxy group is brominated. This is also ortho. It's brominated. Once there is a intermediate is formed like this, at this position, so the lone pair present on this is the C-terminal end of the amino acid group. So this bond pair is coming over here. This bond pair will form a new bond with this carbon. So that time this pi bond is moves over here, and this pi bond come over here, and this H minus picks up this H and going out as a H2. H2 is going out. H2 is going out means it's called dehydrogenation. So you know that dehydrogenation is also called as oxidation. So what is happen here? So there will be a removal of hydrogen. How the hydrogen is removed? A bonded pair is coming over here. This bond pair form a new bond, and this bond pair coming over here. This bond pair is come to this, and this H minus and H. So going out as a H2 as a byproduct, and the obtained compound is having a nature like this. So you know that N double bond C, this nature is there. This C double bond N nature is called oxime nature. So what it will do? So this on hydrolysis. Hydrolysis means cleavage of oxime. So imine also you can consider. C double bond N is called imine. So these imines are sensitive to hydrolysis. So when it is treated with hydrolyzing agent, maybe a what is called as H3O plus. So there will be a cleavage here. So cleavage means this N will go out as a NH2. This C will go out as a CO. So that's what I have written here. A two fragments is obtained. So this fragment will have a colored one. So this can be separated, and one can find out, this can be subjected to C-terminal, means what is sequence finding by Edmonds reagent, and can be possible to know the sequence of amino acid present in this fragment. It is also possible to find out the sequence present in the this fragment. So overall, the cleavage of peptide can be achieved by using uh, hypobromide. So this hypobromide selectively cleaves the peptide bond between the tyrosine and adjacent amino acid. So that's what this is the explanation. Okay, so what is the role of hypobromide? Otherwise, explain the peptide bond cleavage using hypobromide. So then you have to write all these things. Okay, this is about how the hypobromide is used in the peptide bond cleavage. So with this, I will go to the next slide. So next slide, I am explaining about one more reagent I am taking. So that I am explaining. So here I am taking NBS. So NBS is a N-bromosuccinamide. So this N-bromosuccinamide uh, uh, specifically cleaves the peptide bond between the tryptophan. This is called tryptophan and adjacent amino acid. So that means there will be a cleavage here. How it is cleavaging? So that is the mechanism we are explaining here. So NBS, it is a reagent called N-bromosuccinamide. So it contains Br radical. So Br plus, it contains Br plus. That means it will act as a source of Br. NBS will act as a source of Br. So what will happen? You know that this carbon is there, no? This contains and this carbon is electrophilic. What is called when a tryptophan containing peptide is treated with NBS, there will be a new bond formation between this carbon and bromide of NBS. So that is how a compound is obtained. And here in the bond formation, this bond pair is used. So that is the reason this carbon attains a positive charge. So what it means, there will be a new bond formation between this bond pair and Br of NBS. So once there is a involvement of this pi bond in a new bond formation, so then this carbon attains a positive charge. That's what I have written here. Since there is a positive charge, what will happen? This bond pair is coming over here, and there will be a new bond formation between this bonded pair and this carbon. So that is how a imine is formed. So once there is a imine formation, this can be hydrolyzed. So hydrolyzed means this carbon gets converted into CO, this nitrogen gets converted into NH2. So that is how two fragments is obtained. Okay. So what you need to remember, so in the case of hypobromide, tyrosine and adjacent amino acid is cleaved. So in the case of NBS, tryptophan and adjacent amino acid is cleaved. That one you have to remember only the tyrosine, you have to remember only the hypobromide, you have to remember only the tryptophan, you have to remember only the NBS. Just by remembering these two, if you are able to write a peptide, dipeptide, and if you are able to write these steps, then it will be cleared. So normally, so one question can be picked from this concept. So what are the different methods available for the peptide cleavage? So then it will be a 5 marks question. So you can write one method or two method with explanation. So this is about hypobromide, using hypobromide, how the peptide is cleaved. using NBS, how the peptide is cleaved. So hypobromide selectively cleaves the tyrosine and the adjacent amino acid. NBS, tryptophan and adjacent amino acid is cleaved. So here there is a similarity between the cleavage condition. So this cleavage condition is the last step is same in these two methods. So next one I am taking. So next I am taking a compound called cyanogen bromide. I think you are aware about HCN. HCN is called hydrogen cyanide. So CNBR called, it's a cyanogen bromide. So it is also a highly hygroscopic. Hygroscopic means, so if you are keeping the bottle open, bottle lid if you are opening, so that moisture is enough whatever the moisture present in the atmosphere, that moisture taken up by cyanogen bromide and it will releases HCN. HCN is a poisonous gas. So that is the reason handling of this reagent is also a difficult task. And one more thing, the cyanogen bromide at room temperature, it is a liquid, but at lower than the room temperature, it is a solid. So what will happen here in this case, cyanide, Cn minus, Br plus. Okay, that is the condition. So what will happen here? No, 1 minute. In this cyanogen bromide, Cn plus Br minus. So Cn here, it is a positive nature. Br will be in the negative nature. So this, you know that this group, okay, this group is called methionine group. So what it means, cyanogen bromide specifically cleaves the peptide bond between methionine and adjacent amino acid. The methionine means SCH3 containing amino acids are called methionine group. So what will happen here? The lone pair present on the sulfur, it will form a new bond with the cyanogen. So cyanogen bromide. So in this case, Cn will be a positive nature. Br will go out as a Br minus. So there will be a bond formation like this. So you know that there is a positive charge on the sulfur. See, since there is a positive charge on the sulfur, sulfur is a electronegative in nature, then what it will do? It will try to pull this bonded pair of electron. When sulfur is pulling a bonded pair, so that time this bond pair is coming over here. This bond pair, okay, attacks this carbon, okay, and there will be a formation of compound like this. And the last step is same means hydrolysis of imine. So there will be a cleavage here, CO is coming over here, NH2 is coming over here. So this is how the cyanogen bromide cleaves the peptide bond. So now up to this we discussed about what is the cleavage of peptide. So as in acidic cleavage, certain amino acid is cysteine, tryptophan, okay, methionine, these compounds are destroyed because of acidic nature. In the basic hydrolysis method, so amino acids may racemize. So because of that, in spite of that, tryptophan identification is uh, uh, advantage in alkaline hydrolysis cleavage. So in order to overcome this, a special method was introduced to cleave a peptide bond. So that is hypobromide method, HOBR method. So HOBR method specifically cleaves the peptide bond between the tyrosine amino acid and the adjacent amino acid. And then what is called NBS. So NBS, it cleaves the peptide bond between the tryptophan and adjacent amino acid. Cyanogen bromide, it cleaves the peptide bond between the methionine and adjacent amino acid. So these are the methods. By these methods, one can cleave the peptide bond. And so these are all about chemical method of peptide bond cleavage. So now I will go to the enzymatic method of cleavage. So this is the enzymatic method of cleavage. So that means here enzymes are used to cleave the peptide. So enzymes can be used for breaking the peptide bond, and they are called peptidase. So peptidase is an enzyme that cleave the peptides, okay, into two fragments. So that means hydrolysis with the specific peptide, with specific peptide into fragments. Okay, so that means when you are using a enzyme, it is possible to get only the fragment, okay, two products. So not the what is called as individual amino acids. So individual amino acid is not possible with the enzymatic hydrolysis. So by using an enzyme, only one can make a peptide into two fragments. So here, uh, one is endopeptidase, another one is exopeptidase. So endopeptidase means these are the enzymes cleave the peptide chain in between, So that means endopeptidase, these are the enzymes, and these enzymes cleave the peptide chain in between. Okay. See, for example, here I am taking trypsin. So trypsin is an enzyme. It cleaves the amide. So trypsin is an enzyme, more particularly an endopeptidase, and it cleaves the amide where CO is the part of the lysine. So that means lysine, okay, lysine and adjacent amino acid a enzymes cleave the peptide chain either N-terminal N-terminal end or the C-terminal end. So that means endopeptidase or cleave the N-terminal end or the C-terminal end. Is another amino acid, C is another amino acid, D is another amino acid. So that means I took a tetrapeptide. So this tetrapeptide, when it is treated with amino acid, more particularly N-terminal end is cleaved. So that means this N-terminal end is cleaved, and remaining is intact. And one in this example, C-terminal end, okay, C-terminal peptidase, N-terminal peptidase. Here the enzyme is N-terminal peptidase. Here C-terminal peptidase. This cleaves the C-terminal end of the amino acid. So this is about what is called as cleavage of peptides. Okay. So chemical methods and what is called as enzymatic methods. Okay. And peptide cleavage using hypobromide, peptide cleavage using NBS, peptide cleavage using cyanogen bromide. So do you have any doubt up to this? Hello?
No, sir.
So shall I continue a few more hours, few more minutes, so that I can take up the synthesis? So the difficulties in peptide synthesis. To, to overcome the difficulties, a strategy for the, it means key steps. What are the steps involved in a synthesis of peptide or protein? So with this, I will go to the next slide. So next slide will give you the information about synthesis of peptides. So Emil Fischer, okay, Emil Fischer is the scientist, and he first prepared a peptide containing 18 amino acid residues. So with this, he laid a foundation for the peptide or protein chemistry. The basic problem of the peptide synthesis is protecting amino group. Okay, and so what is here it is required? So here it is required one is N-terminal, what you call amino group of the one amino acid should react with the carboxylic group of the another amino acid. So what is the problem here? Suppose a amino group of the one amino acid is react with the carboxylic group of the same amino acid. Then there is a resulting in a unwanted product. So in order to overcome the byproducts or unwanted product, one should think about the protecting amino group. Okay, see for example, okay, for example, in the preparation of glycine alanine. So what is happening here? Amino end of the glycine. So glycine is the N-terminal end, no? And alanine is the C-terminal end. So what is here should happen? Amino group of the alanine should react with the carbonyl group of the glycine. Then only it is possible to form a glycine alanine. Suppose a amino group of the alanine is reacting with the carboxylic group of the same alanine, then it leads to a formation of a peptide called alanyl alanine. Similarly, amino group of the glycine can react with the carboxylic group of the same glycine amino acid. Then it leads to the formation of a compound called glycyl glycine. So what it means? It means that one should proceed simultaneously the formation of unwanted glycyl glycine amino acid, alanine amino acid. To achieve this, okay, to overcome this problem, so what we need to do? So need to do in NH2 group of the starting amino acid, so must be protected by reacting with a suitable protecting group. Then we should know what is protecting group. Okay, so so that I will explain in the next slide. A protecting group is a group so that can be removed later. So protecting group is a group that can be removed later, so without disturbing the any peptide linkage. So that have been built up. So that means protecting group is the one which can be removed later without disturbing the peptide linkage. Okay. Here the protect and after protecting a amino acid means amino group of the amino acid, that protected amino acid is activated. So protected amino acid is I think you know that carboxylic acid group. So carboxylic acid group when it is treated with a amino group of the amino acid, reaction happens and product peptide is obtained. Suppose If we are able to convert a carboxylic acid into a acid chloride, that means COOH to COCl. COCl is the Cl is the better leaving group compared to the OH. That means you can do the reaction at lower temperature. So that is the advantage. So that is the reason here the explanation is protected amino acid is activated. Activated means it is converted into better living group. So by converting amino acid into amino acid chloride by treating with thionyl chloride. Okay, thionyl chloride is a chlorinating agent. The acid chloride is then in separate operation, it is allowed to react with another unprotected amino acid to get a peptide. So this is about what is called as a problem in synthesizing the peptide. And to overcome the problem, one should protect the amino end of means end N-terminal end of the amino group, and after the protection of N-terminal end of the amino group, so it was treated with activation. Acid chloride, acid is converted into acid chloride. So that acid chloride is treated with another amino acid, the one required to make a peptide. So for this peptide synthesis, four steps is involved. So one is protection of amino group. Second one is after the protection, a carboxylic group is activated by converting into acid chloride, and uh, acid chloride, N-protected acid chloride, it is treated with what is called as coupling agent. So coupling means it is treated with another unprotected amino acid, and after the dipeptide or tripeptide or pentapeptide is formed, so deprotection of N protection is made, and now the peptide is ready. So these are the three steps, four steps, key steps are involved in a peptide synthesis. The first step is protection of N-terminal end of the amino group. And after the N-terminal end of the amino acid is protected with a suitable protecting agent, then carboxylic group is activated by converting acid into acid chloride. And the acid chloride, it was treated with unprotected amino acid to form a peptide bond. After the peptide bond, once you got a desired peptide, then you can cleave the N-terminal protected group. So it's called deprotection. So these are the key steps involved in the peptide synthesis. Okay, so what I will do, I will take one example, how the dipeptide is obtained. So that I will explain in the next two slides. So here, so this is the amino acid, and step one, it's a protection of amino acid. It's also called as blocking. So this is the N-terminal end of the amino acid. So this amino group is protected by using a Z group. I think you know that Z. What is Z? Okay, Z may be a Bach. Bach means okay. Anyway, I will take up what is. I think you know. No, Bach. Bach means butyl oxycarbonyl chloride. Okay, and it will react with this NH2 and forming a N-protected amino acid. So Z means butyl oxycarbonyl chloride. So H and Cl is going out, and here there is a formation of bond between the Z and amino. Now the N-terminal end of the amino acid is protected. Now this C-terminal means carboxylic acid group is activated by converting this acid into acid chloride by treating thionyl chloride. So thionyl chloride is the best chlorinating agent because here the byproduct forms are gaseous one. Sulfur dioxide is a gaseous, HCl is a gaseous. Both the things will go out, leaving behind only the N-protected amino acid chloride. This is the second step. This N-protected amino acid chloride, it was treated with unprotected amino acid. So this is R2, unprotected amino acid. Cl and H is going out, forming a dipeptide. So if you want to stop at this stage, you can go for a deprotection of N-protected one. Otherwise, activate this carboxylic group and treat with another amino acid, you will get a tripeptide. And then again activation of carboxylic group, treat with another amino acid, you will get a tetra. So like that, you can go until getting a desired peptide. After that, deprotection of N-protected one. Here, deprotection, removal of protected group, it's called deprotection, removal of this, and obtained one is dipeptide. So suppose R dash is equal to H, then it is called glycine. If R2 is equal to CH3, it's called alanine. Glycyl alanine can be synthesized. So this is about the general method of peptide synthesis. Okay, so what I will do, today we discussed about cleavage of peptides, and also we discussed about peptide synthesis, the problems in peptide synthesis. To overcome a problem, N protection is required, and also we discussed about the steps involved in a peptide synthesis. So if you have any doubts, you can ask me. Otherwise, you can log out. So maybe I will continue remaining part in my next class.